Our first speaker today is Dr. Lily Jan, an HHMI investigator and a professor of physiology at UCSF. Professor Jan is one of the giants in the field of ion channels. Her seminal work on the cloning and characterization of the Drosophila shaker channel, and then its mammalian ortholog KCNA1, is universally regarded as a crucial milestone in the field of ion channel physiology. More recently, Dr. Jan has led the way in the study of TMEM16 chloride channels. Professor Jan is a National Academy member and has a long list of accolades that I will not list here. We are really honored to have her start the symposium with a lecture on ion channel physiology and function. Thank you, Dr. Jan.
Thank you very much. So I will begin the talk by going over the basics about ion channels, how ion concentration gradients are set up by transporters and pumps, how the electrical potential combined with the ion concentration gradient can drive ion flow through channels, how ion channels control the electrical potential across the membrane, how channel activities may be regulated for permeation of certain types of ions, and also how these channel activities can be measured. In the second half of my talk, I will go over a few examples of channel functions that go beyond just letting ions to go across the membrane. That includes volume regulation that's relevant for cell migration and proliferation, as well as examples of multitasking ion channels that have other functions. And thanks to the generosity of my NYU colleagues, Bill and Stefan, I am using actually the slides they prepared for a course this year for the first segment of my talk. So the cell membrane has transmembrane proteins that include ion channels, transporters, and pumps. And because ions cannot readily move across the lipid bilayer that's hydrophobic, we rely on channels for ion permeation across the membrane. And based on the charge carried by the ions, we refer to them as cations or anions. So for example, dissolving calcium chloride salt in water will lead to the generation of calcium ions that are cations and chloride ions that are anions. And calcium ions that can serve the purpose of second messengers by modulating enzymes and channels will have that. So that's the activity we are talking about. But the activity of calcium channels would depend on the concentration, as we all know. So just a few exercises, for lactate, that is an ion because lactic acid dissolved in water will generate lactate as the anion and the proton. And do channels, transporters, and pumps only transport ions? Not exactly. For transporters, some move only ions across the membrane, while others may also transport uncharged molecules. A cell typically has high concentration of potassium inside compared to outside, but high concentration of sodium, calcium, and chloride outside compared to the inside. The membrane bilayer is hydrophobic, so it's not permeable to ions and polar molecules, and the ion concentration gradients that are crucial for cell functions are set up by transporters and pumps. So the ion concentration gradient would tend to be, or I guess I can say that they could be running down because the ions can go through the membrane through various mechanisms. And so we depend on the pumps and transporters to maintain the gradient. And by convention, the current that flows inside into the cell. So what we call inward current, we are considering positively charged ions carrying the current flowing into the cell. And so for current that arise from a net flux of or any positively charged ions outside, from the inside to the outside of the cell, we refer to those as our current. So what about the case of chloride ions moving across the cell? So for chloride ions moving into the cell, we call that the current generated by the inward flux of chloride ions and our current. So that's the convention. I think these are the things that trip everybody up. Those are the jargons that we live by somehow. So for ion concentrations that need to be set up by the pumps and the transporters, it's the ATPase that move sodium ions out and potassium ions in that set up the concentration gradient that's true for all cells, sodium high outside and potassium high inside. And then we have transporters, also known as carriers. So the example I use here is the sodium-calcium exchanger. So transporters don't hydrolyze ATP to generate energy for driving the ions. Instead, they rely on the concentration gradient already set up. So in this case, in this example, it's the sodium concentration gradient that will allow sodium to go in through the sodium-calcium exchanger. And in exchange, this drives calcium ions from inside to the outside to build up the calcium concentration gradient. And we have active transport that's mediated by pumps that hydrolyze ATP to generate energy for the ion transport. And we have passive transport mediated by transporters that rely on the concentration gradient for moving ions across the membrane. The example of sodium-calcium exchanger is one for antiporters because the two types of ions are going in opposite directions. And there are symporters that move different types of ions in the same direction. And for pumps and transporters that cause a net flux of ions or charges across the membrane, they are referred to as electrogenic transport systems. And for pumps and transporters that have no net flow of charges, they are electrogenic. Now, for ion channels that could really allow ions go in either direction, the question is what's driving the ions to move to generate a net flow of charge. So now let's start with the simple example. We have a membrane that's permeable only to potassium ions. And now we change the concentration of on one side. And so when we have the potassium chloride salt dissolved in water, that we end up with potassium ions and chloride ions. So in this example, we will have 5 millimolar potassium ion and 5 millimolar chloride ion on the left side, 140 millimolar potassium ion and 140 millimolar chloride ions on the right side. And since our hypothetical membrane is only permeable to potassium, we just think of the potassium first. We have a concentration gradient that will drive potassium ions from the right to the left. And that will leave an imbalance of charge. We have a net increase of potassium ions on the left and an unbalanced charge chloride ion on the right. And with this, there will be an electrical potential across the membrane that will tend to drive potassium to the right. And so with more and more potassium ions going to one side, we will have a bigger electrical potential across the membrane until the electrical driving force driving potassium to the right is equal and opposite to the chemical driving force from the concentration gradient that will drive potassium to the left. So that's the equilibrium. And so the voltage at this point is the potassium equilibrium potential. We typically just call it minus 90 millivolts. And that's determined by the Nernst equation for potassium equilibrium potential. So typically, potassium concentration is high inside the cell. Can potassium flow into the cell even though the concentration is higher inside? Well, which way the net flow of potassium will be depends on the electrochemical driving force. And that is the difference between the electrical potential across the membrane and the potassium equilibrium potential. And so here, there's another convention that we get used to. We call the electrical potential outside the cell zero. And so the potassium equilibrium potential will be minus 90 millivolts. And the membrane potential above EK, but still relative to the outside will be negative. In this case, we set it at minus 70 millivolts. Now, there are different types of channels on the cell membrane. Here we give a few examples. Potassium channels that preferentially allow potassium to go across, and sodium channels permeate sodium ions. Calcium channels are fairly selective for calcium ions, and chloride channels. And some of the chloride channels are actually permeable to a range of anions. They are usually called chloride channels because chloride is the physiological anion. That's the most abundant one. And besides ion selectivity, the channels may differ in their gating mechanism. Some are voltage gated, so they can only open at a certain range of membrane potential. Some are gated by binding to calcium or calmodulin. Some are ligand gated. They may bind to transmitters, IP3, a range of things. And some may be sensitive to ATP to ADP ratio, so they're metabolically connected, regulated. And then there are some that are activated by stretching the membrane or by changing the volume of the cell. So the membrane potential would depend on the permeability of the membrane to the ions. The physiological ions we typically consider sodium, potassium, and chloride. And the resting cell is primarily permeable to potassium, but it does have permeability to sodium and chloride. And the membrane potential is determined by the permeability of the membrane to these ions and the concentration of these ions on the outside and the inside. So this is the Goldman-Hodgkin-Katz equation. So the plot on the left is an example, just get a sense how the membrane potential would depend on the permeability of sodium and potassium, and typically it's a small number, and also on the potassium concentration. And typically the external potassium concentration is in the millimolar range. And this plot on the right is an example of membrane potential measurement of Kv 1.3 expressing CHO cells, and that's measured at the range of external potassium concentration. So all cells, excitable or non-excitable, have resting membrane potential. They tend to be closer to potassium equilibrium potential because the membrane is primarily permeable to potassium ions. So the graph table on the left shows examples for neurons, skeletal muscles, cardiac myocytes, and so on. And the table on the right shows the membrane potential of human T lymphocytes, different subtypes that are all CD3 positive. But you can see that while they are all very sensitive to the external potassium concentration, the different cell types may have different channel composition. So marker toxin that blocks Kv 1.3 potassium channels will have a higher effect on the membrane potential of memory T cells. So the cell membrane may have many different types of ion channels, and it's those that are active that determine the membrane potential. And if we look at an individual channel, they all have a pore that allow ions to go through. They have a selectivity filter that determines what kind of ions can go through. And then they have a gate that can control the opening and closing of the pore. So at the molecular level, the function of the channel includes permeation, passing ions, and gating. And the example shown here is for a voltage-gated ion channel. Actually, for sodium, calcium, or potassium, it's the same design. There's a pore, and there's a selectivity filter that controls the type of ions that can go through. And then there's a voltage sensor that bears charges that allow the voltage sensor to respond to the changes in membrane potential and control the gate that can open or close the pore. And in fact, if you remove the ions that can go through the pore, you can detect the movement of this voltage sensor by the measurement of gating current, because the sensor is part of the protein that carries charge residues that actually move in the membrane when the potential across the membrane changes. And so, as I mentioned before, we have these different channels that are fairly selective to different types of ions. There are also channels that, so for instance, the ligand-gated ion channels may permeate both sodium and potassium, so they are not very selective, and they often are referred to as non-selective cation channels. Now, given that channels carry ions across the membrane and movement of ions that carry charge generate current, we should go over the electricity. So suppose we connect a battery to the ends of a resistor. So now we have a voltage drop across the resistor, and that will drive charge to move across the resistor. And the larger the resistance of the resistor, the smaller the current that will go across for a given voltage across the resistor. So that's the Ohm's law. Now, if we connect the battery to a capacitor that's made of insulating material in the middle, so the charge cannot go across, and still positively charged ions, or it could be whichever carrier for charge, will be moving away from the positive end, and negatively charged charges will be moving toward the positive end. And that applies here also. And since the insulating material prevents the charge from going across the capacitor, we have a buildup of positive charges on one side and negative charges on the other side. And so in this case, the bigger the capacitance of the insulating material, the more charges will build up for the same voltage across the membrane. So charge is the product of the capacitance and the voltage. So now we go to a cell. So in this case, the battery could be a stand-in for the electrochemical gradient or the electrochemical driving force for the ion, which can go across the membrane through the channels. The conductance of a channel is a measure of how easily ions can go through the channel. So the conductance is inversely proportional to the resistance. And on an IV plot where we measure the current as a function of voltage, the slope of this IV will give a measure of the conductance. Now, as I mentioned, the hydrophobic membrane is not permeable to ions, so it functions as a capacitor that will allow ions to accumulate on either side. So that's a capacitor. And the capacitance of the membrane will be proportional to the area of the cell membrane. And so here we have the charge accumulating on either side of the membrane. That's the product of the membrane capacitance and the membrane potential. And now if we change the membrane potential, we will see a change in the amount of charge that's on either side of the membrane, and that gives rise to the capacitive current. So for a typical cell that will have both the membrane capacitance and the channels that will allow ions to go through, we have any current measured across the cell membrane to be the sum of the ionic current and the capacitive current. So with a voltage jump, we will see first the capacitive current and then the ionic current that depends on the conductance of the channel and the driving force, actually. So how to measure the channel activity? We measure the current, and there are different ways. There's the voltage clamp mode and the current clamp mode. And with current clamp, the example shown on the right, we measure the voltage across the membrane with or without the injection of current. So in this example, there are different amount of current injected into the neuron, and that will lead to the firing of action potential. For voltage clamp, we hold the membrane potential at a constant level. Here there are multiple different superimposition of different membrane potentials. And when we bring the membrane potential to a different level, that might cause the channels to open, allowing ions to flow through the cell membrane, and that would tend to change the membrane potential. So we use a feedback circuit to inject current into the cell, balance out the current flowing through the channel to maintain the membrane potential at this constant level. And we measure the amount of current injected into the cell via the feedback loop, and that gives a measure of the current flowing through the channel. And we can use different kinds of electrodes. We can use sharp electrodes that penetrate the cell membrane, or we can use patch electrode that forms a tight seal with the membrane. And so these are direct methods. And I will also get into indirect methods that measure the channel activity by monitoring ion fluxes or monitoring the fluorescence of sensors for voltage or calcium. So for a cell that's large enough to allow sharp electrodes to go through, we have 2 electrodes in the cell. We have one electrode that sets the membrane potential at this command potential that we specify. And if that leads to opening of channels and flowing of ions through the channel, that would tend to drive the membrane potential away from the command potential. And any deviation from the command potential will be detected by the feedback circuit, and that will lead to current injection into the cell in order to maintain the membrane potential constant at the command potential. So the current injecting into the cell gives a measure of the current that's flowing through the channel. For patch electrode, that makes it possible to record from small cells like lymphocytes. And in this example, we have a single channel that's active in the membrane patch that's in the, so once you have a tight seal, you have a membrane patch that will be, if the patch is still there, any channel that's open, the current flowing through will be detected by the electrode. And we can actually use the same electrode for voltage clamp. So the same electrode will measure the potential across the membrane patch and also pass current via the feedback circuit to maintain the membrane potential at a constant level. And so in this case, the currents flowing through, via the feedback circuit, it gives the measure of the single channel current. And this is one example where the current will change direction somewhere here in between minus 20 and plus 20 millivolt. And that's an example for a non-selective cation channel that permeates both sodium and potassium. And so this reversal potential, it's somewhere in between the sodium equilibrium potential and the potassium equilibrium potential. The way it really looks like is shown here. The example on the upper right is the patch electrode forming seal with a cardiomyocyte. And you see that with an inverted microscope. And there are manipulators holding the electrodes that you can move around. And there are perfusion systems for changing solutions or applying compound to the channels you are recording from. So there are different modes for patch recording. Once the patch electrode forms a tight seal with the cell membrane, we can do the cell attach recording. That's the example I showed you before. Or we can pull off this patch of membrane so that the cytoplasmic side of the channel is exposed to the solution. So it's easy to change the cytoplasmic solution and applying any modulators. Alternatively, you can break off this patch so that now you are actually recording from channels on the entire cell membrane. And the solution in the pipette is continuous with the cytosol. You could also, so this is the whole cell recording. You could also do outside-out patch by pulling the membrane and allowing it to seal so that the external side of the channel is exposed to the solution. So that in this example of inside-out recording of an ATP-sensitive potassium channel, we can see the channel activity. And applying ATP to the cytoplasmic side of the channel will inhibit the channel. And so we see now very little activity. And this is an example for whole cell recording. And the current here is the sum of all the currents through different channels on the cell membrane recorded with voltage clamp mode. And it's also possible to monitor calcium in the cell concurrently with patch recording in whole cell mode. In this case, the cells are loaded with the calcium dye flow 4, and so we can see the calcium imaging at the same time as recording with the patch electrode. And it's also possible to have automated system for patch recording and automated system for monitoring calcium in many different wells. And also another automated system will measure the ion flux for potassium. That could be thallium. And if you are doing this automated system for hundreds of wells, you can screen small molecules and look for modulators that increase or decrease the channel activity. So to recap, the basic idea about ion channel function concerns ion flow that's influenced by the membrane potential. The ion flow across the membrane in turn will change the electrical potential across the membrane. Ion channel activity also plays a role in other functions that goes beyond just movement of ions to change the membrane potential. So the example I include here is volume regulation. A change in the volume of the cell involves a movement of water, moving water into the cell to increase the volume, or moving water out of the cell to decrease volume. And the water movement is driven by osmotic gradient that involves movement of ions. And this could be mediated by volume-regulated channel, like this case, volume-regulated anion channel. Or in addition, it could also involve changes in the number or the activity of potassium channels and chloride channels. And for the case of increasing the cell volume, it quite often involves changes in the activity of transporters, like the sodium proton exchanger, and these will transport their activities will drive sodium into the cell. For cell migration, there's actually local volume change. So the leading edge may have local volume increase, while the trailing edge may have volume decrease. And that may depend on the activity of potassium channels. In this example, it's the EAG2 potassium channel. And there are other functions for migrating cell. For instance, the potassium channel activity could change the membrane potential. So here's another jargon. Hyperpolarization means making the membrane potential more negative. And remember, the outside is 0, so the inside is negative. So let's say making it from -75 to -80. And so this would increase the electrochemical driving force for calcium and therefore increase calcium influx. And calcium, being a second messenger, may mediate processes like modulation of focal adhesion turnover. Now, looking at the site of focal adhesion, there are other channels like the mechanosensitive piezo channel that may respond to changes in the stiffening of the tissue by adjusting calcium signaling for remodeling of extracellular matrix. That will be a feedback regulation. And it could also modulate processes that affect cell proliferation. For cell proliferation, in this example, we see one type of chloride channel and the EAG2 potassium channel that mediate volume change actually at S2. It's referred to as pre-mitotic condensation, and that is important for one checkpoint for mitotic entry. And there are other potassium channels that control the volume during mitosis. So now getting to the last point about multitasking channels. The first example I will use is a voltage-gated potassium channel. Kv 2.1 is found at the contact site between plasma membrane and endoplasmic reticulum. It can bind to VAP, that's VAMP-associated protein. And there are multiple levels of modulation. So for one thing, the channels at this position, they are clustered together. They may actually not pass ions, but their voltage sensors may still move in response to membrane potential change. And that's based on gating current measurements. And these ER plasma membrane contact sites are located close to calcium channels and also to close to traffic hub for channel insertion and also for exocytosis involving dense core vesicles. This is an image of Kv 2.1 with these clusters corresponding to the ER plasma membrane contact sites. And this is sensitive to neuronal activity. So glutamate application that leads to burst firing of action potentials would cause these clusters to be dispersed. And the underlying mechanism likely involve calcineurin dephosphorylation of Kv 2.1, causing the dissociation of Kv 2.1 from VAP and leading to the dispersion of Kv 2.1 on the cell membrane. Now, the last example I will use about the multitasking channels is TMEM16F. It's a calcium activated ion channel, and it's also a calcium activated lipid scramblase. And the lipid scramblase activity that allows phosphatidylserine that's normally on the inner leaflet to be exposed to the cell surface will facilitate the fusion of these placenta cells. So that will lead to the formation of the syncytial trophoblasts. So we can see the physiological importance of this by looking at the TMEM16F knockout mice. This is the study of Huanghe Yan's group at Duke. So the knockout mice will have smaller placenta and also smaller mouse embryo. TMEM16F is also important for cell fusion in pathological conditions, like the lung of COVID-19 patients. Prompted by the finding of these syncytia that reflects the fusion of lung cells that are positive for spike protein, Braga and colleagues at in UK and also in Italy developed cell fusion assays and they show that either pharmacological inhibition or knockdown of TMEM16F will reduce cell fusion. TMEM16F also helps with fusion between host cells and HIV virus. So in this case, the envelope protein interacting with the co-receptor leads to increase of internal calcium concentration, and that causes activation of TMEM16F and the flipping of lipids, scrambling of lipids, exposing phosphatidylserine on the surface, and that facilitates the fusion of the virus with the cell. So these two examples of multitasking channels actually belong to two very different, structurally completely different families of channels. And you will hear from Stephen Lung's talk after mine, the structure function studies of different types of ion channels. And quite often, the first few members of a family receive a lot more attention, while other family members are under study. So that prompted this program called the Illuminating Druggable Genome that's sponsored by NIH to generate resources that will make it easier for researchers to examine the functions of these channels. The same program actually includes also kinases and GPCRs. So I'll be happy to answer any questions about ion channels or about this program. Thank you very much.
Thank you, Dr. Jan. I haven't seen any questions pop up yet on Q&A. So while we are waiting for that, I wanted to ask you something about your own journey. So you started with shaker channels in Drosophila. So I imagine you were not a trained physiologist when you bumped into shaker channels. So how did you navigate that early phase, which I imagine could have been daunting for you, into electrophysiology?
Right. Actually, what happened was we went the other way. So at the time, and I were both postdocs with Seymour Benzer. And so we actually started, speaking of Woods Hole and Cold Spring Harbor, we started the project as a course project at Cold Spring Harbor, a summer course. And so the starting point was a question whether genetics could be useful for neuroscience. It was obvious genetics was crucial, for working out the biosynthetic pathway. And so we were wondering whether that could also work for understanding the synapse, for example. And so we started at the summer course recording from the larval muscle, neuromuscular junction. And that turned out to be a fairly quick experiment because the advantage of the tiny little fly is a larval muscle is isopotential, so you don't have to move around, look for the synapse. Anywhere you poke, you see the synaptic potential. And we use that as the screen to go through behavioral mutants, figuring if we are looking for mutations that affect the synapse, either we get a dead fly or we get something that cannot move normally. And so that's how we came across shaker.
I see.
And then we have to learn how to do the molecular work.
I see. It was a reverse for you.
Yeah.
There is a question on TMEM16, and people are wondering if there are good inhibitors for TMEM16 to look at their function. And as you answer that, perhaps you can also take it more generally, and how should one approach small molecule inhibitors of ion channels? As physiologists, we know that things that can work in electrophysiology may not be the right things to use for cell-based assays. And so how do you have general guidelines for people to how to approach inhibitors, especially for immunologists who may want to try and do some pilot experiments with ion channel blockers?
Sure. So TMEM16 is actually a good example to touch upon this question. Because we started with TMEM16A, that's a calcium activated chloride channel. And there's quite a bit of interest in finding actually both activators and inhibitors for TMEM16A because activators conceivably could help with cystic fibrosis, and inhibitors might help with conditions like diarrhea. And so there have been multiple small molecule screens, and you can do that with the type of high throughput screening. For chloride, there are now YFP mutants that are more sensitive to iodide. So that's used a lot. And in fact, there are activators now being tested for cystic fibrosis treatment. So now, so now I get to the more difficult question. So for instance, for the cell fusion, it would be important to search for inhibitors. And actually the paper I just referred to, that's a paper that just came online in Nature. They did a small molecule screen to look for inhibitors of cell fusion and came to niclosamide. It's one of the FDA approved drugs in the libraries they screen for. And that took them eventually to TMEM16F. And so in this example, niclosamide blocks both calcium activated chloride channel, TMEM16A, and TMEM16F. And so, the next step, I think, would be looking for more specific modulators. You could just start with one modulator and do the structure activity type, or you could do it more extensively. And what we have been trying for years, actually, together with Brian Shortcut, was the idea of starting with in silico docking. And if that works, and Brian Shortcut had the Zinc library as a virtual library that I think now it's possibly more than 1 billion chemotypes. And so the hope is the more diverse and numerous the chenotypes represented in a library, the better chance of identifying a modulator that really fits the channel you are interested in. This has worked really well between Brian Shortcut and Brian Ross for looking for modulators of G protein coupled receptors. Mm-hmm. So it would be great if it also works for channels.
Yeah. There is one question which I think is pertinent to immunologists. So oftentimes people will find expression of voltage-gated channels in immune cells. Now we know that sodium and potassium, voltage-gated sodium and potassium channels could have genuine roles in non-excitable cells, but also there are also voltage-gated calcium channels. So do you Can you summarize for the audience how voltage-gated sodium and potassium channels could affect calcium signaling, for example? And perhaps if you have any views on what we might think about expression of voltage-gated calcium channels in T cells, for example.
Right. So all three types are structurally very similar. They are in the same family. I'm going in the wrong direction. That's this one on the left. And so and you can see it's a huge family, and different family members may have clearly there are variations in terms of the voltage dependence. So we call them voltage gated, but some of them may have activity at the resting membrane potential already. And so you may not need a cell that fires action potential to activate, to have active voltage gated ion channels. And so for any channel that's active with an open pore, then how much ion flux, net ion flux. So for calcium channel, the net calcium influx will change the calcium concentration and change the second messenger activity. That can be modulated by the electrochemical driving force for calcium. And part of it is the electrical driving force, right? So if the cell has very active potassium channel, it will drive the membrane potential toward potassium equilibrium potential, more negative than the resting membrane potential. And that will mean a bigger driving force for calcium to go into the cell.
Yeah. I have a pertinent comment from Michael Cahalan, and he points out that a lot of voltage-gated calcium channel blockers can block Kv1.3, and that has often been used as evidence of functional significance of voltage-gated calcium channels in T cells. So I think this, perhaps you can comment on how much, what kind of specificity studies people can do when they look at inhibitors, because obviously we deal with this all the time, that many of these inhibitors are often used at high concentrations, and they may not actually work as intended.
Right, it's good point. That Michael points out, because we sometimes really rely on the pharmacology, and so. It's a difficult question. Obviously, you cannot test any modulator against all channels and other molecules. It may have some effect, but certainly very important to test potentially relevant channels that's in your system. I guess that might involve heterologous expression, just testing them one at a time.
Yeah. I think this is where perhaps immunologists should consult an electrophysiologist and approach these studies a little bit with more insight, so that they don't start building up data without perhaps getting a full grasp of the pharmacology.
And just like any pharmacology, right?
Yeah.
Would be better if multiple inhibitors have the same effect. Multiple inhibitors that are not structurally related.
That's right. What are your thoughts on ion channels in organelles? There has been a lot of activity in intracellular organelles, lysosomes, mitochondria. You actually showed some slide on mitochondrial ion channels.
Right.
How do you see, how have you been following that work, and what are your thoughts on it in general?
So, channels on mitochondrial membrane, channels on lysosomal, endosomal lysosomal membrane, and channels on ER membrane all have important functions. And there are ways, not many labs have that expertise to do it, but it's possible to record from channels in these intracellular compartments. Right. And that would be, I guess, the most direct way to assess the function. And then getting to the multitasking, we actually have contact sites between ER and all these organelles, mitochondria. And there are examples. So for instance, TMEM16K is at the contact site between ER and late endosome lysosome membrane. And it's a scramblase, most likely. We don't know the channel function because nobody has gotten to the point of recording from that.
Right. There is a question on whether people are trying to develop TMEM16F drugs for anti-HIV or some kind of antiviral treatment.
Right. So it was a challenge. So early on, so we collaborated with Ming Li when he was at Hopkins, before he went to GSK. But he was running at the NIH small molecule screen for channels, running the center. So our effort for looking for calcium activated chloride channel, TMEM16A modulators, was done together with Ming Li's lab at Hopkins. And we talked a lot about how to look for TMEM16F modulators. It's not impossible, but it's a lot more difficult than TMEM16A because for TMEM16F, it requires both depolarization, making the membrane more positive on the cytoplasmic side, and calcium to activate. And that just makes it more difficult for the high throughput screen. And so far, like niclosamide, it's testing compounds that worked on TMEM16A to see if it also affects TMEM16F. But down the line, if it's possible to, for instance, using Brian Shortcat's approach, the way Brian Shortcat and Brian Ross pursue the zinc library has been, they will identify in silico, the top 20 hits and test it on a panel of structurally related GPCR. So, and then go through it 5 rounds. So each time testing the top 20 hits. So they hope, and they manage to improve the affinity, but they also have some idea about the specificity of the hits each round. So that will be what I think is doable if we can get off the first step. Yeah.
For those who want to explore TMEM16F in immune cells, and we know that it's expressed in immune cells, you actually published on its role in host defense in Drosophila. How might they approach it using small molecules? Do you have any recommendations for which small molecules are ideal to look at them in cell-based assays for TMEM16?
Right now, it's the niclosamide family that seem to be, inhibiting both 16A and 16F. And I should also mention, for those who are really interested in looking into TMEM16F in lymphocytes, there are other functions. So TMEM16F is linked to the bleeding disorder, the Scotch syndrome. The reason is when we get a cut, the platelets are activated, calcium increases, and that leads to 16F activation, PS exposure, and then the whole protease cascade to generate thrombin, right? But it also leads to microvesicle formation. And that's true not just for platelets, but also for T lymphocytes and B lymphocytes. And so that's another function of 16F in these blood cells. When they expose PS, they leads to microvesicle formation. And And that's a whole nother field. What's inside the vesicles may depend on both the cell type and the way they are stimulated, and what they do once they come, get released.
And I think we are drawing to a close. So as my last question, I know you published the role of TMEM16 in Drosophila host defense. That was some time ago. Have you guys done any more work in it in that direction? Any new insights?
So we're trying, but not really on the host defense angle. And I should also refer you to paper from our former lab member, Huang He-Yan. He has his lab at Duke, and he looked at the Drosophila subdued, the TMEM family member, and he thinks that this one also is a scramblase.
Mm-hmm. I see. Great. Thank you so much, Dr. Jan. That was really great, and everybody really enjoyed it.